I'm sorry, I apologize. So I completely forgot. So we have Dan's for dinner today. So Dan Burns is taking for dinner. Anthony took him last night. So if anyone wishes to join them for dinner, just come after the seminar and talk to Dan's. So we can agree the time and then go for dinner all together. And tomorrow after 10:30, there are opportunities to meet him from 10:30 to 1. So it's 11. No, it's from 11 to 1. So from 11 to 1, there are opportunities to meet him. So if you like to meet him tomorrow, so just let us know. Thank you.
It'll be interesting to see if there are any takers. I have to start out by acknowledging my funding. These are the people that have contributed to the work that I've done, and these are some of the collaborators. And I'm not going to take time to read everything. I assume you're all speed readers. The goals for this talk are to build a case for why pursuit of mechanistic models that better explain biological phenomena require new classes of models. I'm going to describe the approach that I've been thinking about for building such models. At the end, I'll provide some snapshots of our work. And a big goal is to try to encourage you to explore and contribute to this important new frontier. This is not an exercise for one person by any stretch. In order to really reach the vision that I and others have, it's going to take a lot of additional work. And in the process, I'm going to present several heretical ideas for comment, just to make sure that you're all not sleeping there. I found these 4 quotes in the Cambridge Companion to the Philosophy of Biology that I liked very much. I thought I'd share them with you. I'm sure you've read this first one already. This other one, biomedical science can be characterized as the pursuit of mechanistic models that better explain biological phenomena. Advances in biomedical science require more explanatory mechanistic models. That's what I'm interested in. Models that do a better job of explaining the phenomena that we're interested in. And finally, science can be defined as the pursuit of better models. How many people do a lot of wet lab work? When I poll people that do wet lab work and ask them, what fraction of the experiments that you initiate actually wind up being fruitful in terms of generating the new knowledge that you anticipated, helping you make a decision that you needed to make, is publishable in some way? I get a range of ratios, but 1 in 10 is kind of at the low end of the ratios. That's not very good productivity, 1 in 10. 1 in 10 experiments being fruitful. Why is it 1 in 10? I think that even today there is an over-reliance on mental models that are always flawed in ways that only become clear after conducting new experiments. And those experiments could be either wet lab or in the future in silico experiments. I envision that the use of new and established modeling and simulation methods near term, next 10 years or so, should be able to help get that number down from 1 in 10 to maybe 1 in 7. That's a good gain in productivity. We need that gain. By the time your students are halfway through their careers, I believe that we'll be down to 1 in 5. And at that time, if it goes down to 1 in 5, whenever it does, then the entire biomedical R&D landscape will have changed. Things will be completely different than they are today. You'll need half as many wet lab scientists and 10 times as many computational scientists. But seriously, things will be better because science will be progressing on a more efficient basis. It will be standard operating procedure to do many simulated experiments in advance of wet lab experiments. So I try to tell the graduate students that the path they're on is to become expert in designing and conducting in silico experiments. This is very disheartening. This shows the amount of money spent by the larger pharmaceutical companies over the past 10 plus years, 11 years, and the productivity that they've experienced. That's very discouraging. and that trend continues. It can't continue. Obviously, something is going to happen. Either the productivity is going to turn around, maybe by the help of computational models, maybe not, or the model, the business model will collapse. And if it does, it'll create a whole new area of entrepreneurial activity. And the in silico aspect of that, no matter which way it goes, is going to be very interesting. This is a quote that I found from someone who is lamenting the problems with the pharmaceutical industry. And of course, those problems are mirrored over in the academic research sector. They spend less money, but they're less productive than in terms of efficiency than the pharmaceutical industry. And one of the reasons is that back in 1970, there were more low-hanging fruit. The complexity of the issues that had to be dealt with by the scientists that were making the decisions was just less than it is today. But even with this flood of data that we have and the scientific advances that have happened since 1970, the productivity has continued to go down. So the productivity in terms of omics, data, new methods, etc., is not helping as much as we had anticipated. Omics data have not lived up to expectations. So what is the obstacle? To be more useful, I think that the omics data needs an explanatory mechanistic context. So when I talk with colleagues that have that data, we're looking for how we can merge mechanisms with that information. My take-home messages are as follows. I believe that discovering and developing new therapeutics requires acquiring and exploiting deep insight into the generative mechanistic networks that are responsible for the phenomena. And that doing so in the face of incomplete data, it'll always be incomplete, multiple uncertainties, there'll always be multiple uncertainties, requires building and challenging better mechanistic models. The methods outlined in this talk can, I believe, facilitate accrual and validation of mechanisms, help form concrete hypotheses for translation, bring into focus concrete details for particular interventions, and increase the ease with which accumulated mechanistic knowledge is measured, visualized, and leveraged. I found this little diagram to be very useful in talking with all manner of colleagues. These 4 scales or spectra can be used to describe any particular current research problem. And what I do when I'm talking with wet lab colleagues is say, let's define the problem and then tell me where on these scales you think you are in terms of information. Measures can be precise analytically, and yet they can be imprecise in terms of the information that they're giving. You might be in a situation where a certainty prevails or uncertainty prevails. The system information can be complete and detailed, and by that I mean the fine-grained explanatory mechanistic knowledge that explains the phenotype that you're interested in. It can be complete and detailed, or it can be sparse and vague. The observation and phenomena, all of them are as expected, or occasionally some of them are anomalous, surprising, puzzling, or new. Those of you doing wet lab work, where are you? I think the answer to that question determines how you should think about models. Where you are on these various spectra, I think determines how models, what models you should use, and how those models can best be used. And I found it very helpful to approach it from that direction. If I'm on the far right, I might be doing biophysics problems, working on enzyme, protein structures, classical engineering, working with materials. If I'm center left, I'm probably dealing with actual biomedical problems. All of the biomedical research people that I've talked to tend to think that they're center left on at least one of the lower 3 scales. Looking at the systems information model On the far right, the mechanisms, the generative knowledge to explain a phenomena is known, and the mechanisms are actual. And on the far left, they're possible, and then you have in the middle, they're plausible or likely. My notion is that it's very unlikely that biological mechanisms are ever likely, but they can be quite plausible. On the uncertainty scale, certainty can be complete and uncertainty can be pervasive. Wet lab data is interesting. It creates some interesting problems for modelers, both in terms of uncertainty and in terms of variability. If we think about quantitative mappings from wet lab to wet lab, and I'm talking about precise quantitative mappings. If I'm measuring the metabolic clearance in vitro of a compound in hepatocytes, and it's so many micromoles per minute per cell per whatever, what are the chances that that precise measurement, if I take the same compounds and the same cells and do it 3 months from now, are going to match 1-to-1? Anybody want to bet on that? So if biology experiments don't map 1-to-1 from wet lab to wet lab, even for the same experiments, why do you insist that a model map 1-to-1 quantitatively to any particular research result? Just a question. These are two disturbing papers. 3 large pharmaceutical companies over the past 10 years have made it standard practice to actually replicate the experiments that are about to inform important decisions, or experiments that are trying to inform decisions that have already been made. And they found that, and all of the papers came from the best journals. Less than 25% of the work was reproducible. That's not a criticism of the scientists. It's not a criticism of the methods or anything like that. It's just a fact. Biology can be hard to deal with. And this paper talks about the perspective from the venture capital point of view. A lot of people bring some very interesting information to venture capitalists and saying, I have this and it'll do that. And the rule of thumb is don't believe any more than half of it. And the half that you do believe, go out and independently have it verified in some way. So there's actually been a kind of a growth in terms of small labs, independent labs that do work for venture capital firms in terms of replicating wet lab experiments. So this is the reality that we need to be able to work in order to do models, good models. Why is this the way it is? I do a lot of work with hepatocytes, or my current wet lab colleagues do a lot of work with hepatocytes. And they, for example, engage simultaneously in dozens, if not hundreds, of functions that share pathways and components. Does it make sense that a set of hepatocytes in culture are going to be behaving the same way on hour 1 as they are on hour 3? Or that the hepatocytes isolated from 2 different rats from the same strain are necessarily going to be in the same states? I don't think so. The relative preponderance of those functions, coupled with their recent histories, need not be the same in 2 sets of hepatocytes isolated similarly from different labs from the same age, sex, and strain mice, for example. It's hard to deal with that from a modeling perspective. Consequently, the mechanistic cascade that might unfold with an intervention may simply unfold differently in the 2 sets of hepatocytes. The biology, too, can change. Hepatocytes change upon isolation and stabilize differently depending on the isolation method that you use and the culture conditions that you put them in. Thus, it's not very surprising that there are few 1-to-1 quantitative correspondence between comparable measures taken from different experiments having the same focus. Yet, how many articles do you read about high throughput experiments? That is true even when the protocol focuses on exactly the same phenomena, which is the case for high throughput experiments. Consequently, many researchers, the wet lab researchers, place far more trust in the relative phenomena details than in the absolute quantitative measures. They place more trust in variable to variable comparison rather than value to value correspondence. And that reliance is reflected in both published and experimental designs and the vast majority of published mechanistic descriptions. So that reality, having lived it, has kind of influenced the way that I'm trying to come at modeling and simulation, because what I would like to see in models is a situation that mirrors what's going on in the wet lab. And yet I still want to use them to make better decisions. So if I think about predictions and line them up with my graph, on the far right, I might be able to make trustable, precise predictions for a new context. So if I'm breaking a steel rod under different experimental conditions, I can make accurate, trustable predictions about how that will break. But how about a new intervention on cultured cells? At the other end of this prediction spectrum, the best I can do is just an informed opinion. Most of the decisions made in pharmaceutical companies are based on the informed opinion of experts, even in the face of experimental and computational observations. And depending on where we are on the system information scale, that maps up into the predictability scale and tells us something about what we might be able to predict or not predict. On the right-hand side of, if I'm on the right-hand side of all the scales, I can make precise predictions. If I'm on the left-hand side of one of the three bottom scales, I think that the best, what you should focus on is improving your description of the system, which means discovery and exploration, exploring mechanism space and discovering incremental, more plausible mechanisms. We'd like to move from some location on the system information scale incrementally to the right. In other words, we need models that help us shrink the space of plausible explanatory mechanism down. And that automatically says that, that we're always going to be dealing with many plausible mechanisms for any particular phenomena that we deal with. So our models need to be able to simultaneously represent many plausible mechanisms instead of just one. For any particular experiment, a model is going to have a corresponding use case or in silico experimental use. I don't know how many of you are comfortable using the term use case, but I just want to define it because I use it quite a bit. The use case is the aspects of the referent that the analog system is intended to mimic. And I'm switching now from the use of model to the use of analog, because the systems that I deal with are, as you'll see, more analogous to the biology than they are models of the biology. If we're center left on any one of the three lower scales, I think it's hard to model the biology because we don't know enough to do it, but we can mimic the biology. It's how and for what purpose the model will be used. Simulation scenarios, the component or analog's phenotype, the set of targeted attributes. That's a use case. So I want models that have use cases that match to the experimental use cases in the wet lab. On the right-hand side of these four scales, continuous mathematics is perfectly appropriate. We have a solid, deep foundation in those mathematics, and those formalisms are well encapsulated, and they work best when we are absolutely on the right-hand side of all four scales. If we insist on using those methods when we actually are living center left, we have to make a lot of assumptions. a lot of simplifications, and the more complicated the model becomes, the more complicated that daisy chain of assumptions becomes, and it creates lots of problems. So on the left-hand side, the only thing that I've been able to see that we can do right now is to focus on the use of object-oriented, agent-oriented, and actor-based models. And I'll say a little bit about those in a minute. But we can have mixtures of the two types depending on where we are on these various scales. So I envision relying on analog models if we're center left. And I define an analog as anything that is analogous or similar to something else and that exists and operates in isolation, even in the absence of a referent. It's a system that has aspects and attributes that are similar to those of the reference system. And in the biological context, it's biomimetic software that, when executed, produces phenomena that mimic those of the analog reference. That's what I mean by an analog. When in pursuit of explanatory mechanistic insight, I think that the emphasis should be on generation, exploration, and falsification of multiple mechanistic hypotheses. But we're still in a space of lots of plausible explanations for any one phenomena that we might see. And to populate and shrink the space of plausible mechanistic hypotheses, I think the focus on should be weakly on validation, but more importantly on mechanism falsification. Falsification gives us new knowledge, validation and verification gives us no new knowledge. And what we want to do is take that space of plausible mechanisms, falsify one of them, and shrink it a little bit. And the more that we can pressure that space of plausible explanations, the more useful knowledge that we'll get. What analog capabilities would be needed in order to do something like that? I'll list 6. Reusability. The analog components can be reconfigured to represent different mechanistic hypotheses and even different wet lab systems. If I'm going to be exploring mechanisms, trying to make the mechanisms more realistic, I'm going to be constantly changing and altering my model. So I need to have components that can be reused under different conditions. The components need to articulate. That is, it's easy to join, disconnect, and replace components both vertically and horizontally without having to significantly re-engineer the analog. That's hard to do with a set of equations. I would like to have a model where it's easy to do because I know that I'm always going to be constantly in the process of revising and altering and improving my mechanisms. By articulation, I'm talking about the extent to which the analog consists of distinct, interconnected parts or components, and the extent to which these components are encapsulated and their internal dynamics are independent of those of other components. I envision a lot of biology consisting of encapsulated components, and the components within that encapsulation being somewhat independent of each other. Flexibility, it's relatively simple to increase or decrease granularity, and that doing so enables cycles of hypothesis testing, falsification, and refinement. Local mechanisms. Simulated phenomena are a consequence only of local mechanisms. In other words, I don't infuse my opinion about what's going to happen in the mechanism into the mechanism. I allow the mechanism to unfurl by allowing components to interact with each other only locally. They don't use information that's not local. Transparency. One can observe and measure simulation details as they unfold and compare them to measured referent wet lab phenomena. And finally, measurable experimental similarity. The goal should be to, as I see it, the goal should be to have an analog that, when executed, produces phenomena that's measurably similar to the phenomena over here in the wet lab. And I'll talk more about that. These 6 capabilities require analog components that rely primarily on relational grounding. And that's a kind of a term I think that maybe I invented. And I'll just say a little bit about grounding because I think grounding determines an awful lot about how models can and should be used. And this is one of the heretical topics, causes a lot of my modeling and simulation people, friends, to get very upset. Grounding is the units, dimensions, objects to which a variable or constituent refers or relates. Absolute grounding, all the variables, parameters, and input, output are in real-world units like seconds and meters. In relational grounding, the variables and parameters are in input, output in terms of defined by other components in the model. For example, if a component output is in a set of form a lumen, elongate, bifurcate, branch, form a clef, and the receiving component accepts those elements, then they're relationally grounded to each other. I claim biology uses relational grounding. So if biology is using relational grounding, maybe we should put more effort into using relational grounding in our models. Grounding and its impact on analog use can be commented on with a couple of following observations. Inductive models typically rely on absolute grounding. We had a good conversation earlier about absolute grounding of inductive models. Absolute grounding can create an entangled daisy chain of often weak assumptions. Absolute grounding complicates combining models to form larger ones. Relational grounding enables flexible, adaptable analogs, but it requires a separate analog to referent quantitative mapping model. I don't think that's a problem. I actually view traditional equation-based models as a conflation of 2 models, one describing a phenomena and the other describing the relationship of that phenomena in terms of units. So it's two models conflated into one. So I envision a grounding scale that goes along with these other 4 scales. And that on the right-hand side, I'm perfectly comfortable using relational grounding. And if I'm way over on the left-hand side, I claim that you should focus on using, I think I said it backward. On the left-hand side, I claim that you should focus on using relational grounding. And in the middle, you might, depending on where you are on the other scales, you might use a combination of grounding methods. But the location on those scales would inform you as to how you should think about these grounding issues. The use of the analogs that I'm describing and their functions are fundamentally different conceptually from the use cases and functions of conventional equation-based models. I'm interested in increasing phenotype overlap and increasing mechanistic and phenotypic similarity. And I'd like to just take a couple of minutes and just talk about some of those ideas. The asterisk indicates a phenotype, an attribute of the system that I'm interested in. We'd like humans, animals, in vitro systems to have a lot of phenotype overlap, but we know they don't. So I automatically have a mapping problem between the attributes of interest in these 3 systems. Under some really good circumstances, we might find a situation like this, where there is some attribute overlap, some phenotype similarity. Nevertheless, what I would like to do is to be able to build analogs that have their own phenotype, and that phenotype can overlap a little bit with the phenotype of whatever the reference system is. And that with a little iterative improvement, trying to do more in terms of quantitative matching, I can actually improve that. The phenotype of the analog, however, will never completely overlap the phenotype of the wet lab model in the same way that the wet lab model phenotype doesn't overlap the phenotype of another wet lab model. The nice thing about analogs of the type that I'm talking about is that if I have 2 analogs that use more or less the same components, and they overlap a little bit with two different biological systems, as indicated here, I can put a software wrapper around those two models and explore the space of morphing between one analog and another. In other words, I could ask the question, what is gained and lost in my analog when I morph it and make it transform into the analog counterpart of the mouse, and vice versa? What I have to throw out and add in order to make the mouse analog more like the in vitro analog. And in exploring that software analog morphing in silico helps me think more, helps me think better about the corresponding process in the biology, which I can't actually explore. And in time, I'd like to be able to do this. I'd like to have analogs that have undergone enough validation and falsification and so on and so forth that morphing method would actually allow me to say something about what's happening in people and have a degree of confidence in that statement. It could count as a prediction, but it's more like a projection. I'm going to say a few words now about how these analogs might increase their mechanistic and phenotypic similarity. This is a sketch of what might happen within a mouse when I give it a drug. We know that the drug eventually finds its way to intracellular components. There's some type of interaction. That interaction propagates to the cell and the tissue level, and that causes things to happen in the case of the liver at the lobule level, and that propagates up and eventually shows up as system-wide effects in the mouse. That's the system that I'm interested in. I want to understand it better so that I can better anticipate the consequences of interventions. So I'd like to have an analog where the major components in each of those systems have counterparts in the analog. And I'd be very interested in improving the mapping between those different components. The mapping could be improved qualitatively, quantitatively, or in terms of behavior. In order to improve that mapping, I have to focus a little bit on measurement similarity. For example, I need to measure things that are occurring in the analog and compare those to measured things in the wet lab system. And I need to identify a similarity that I'm going to be happy with. It's best if I can identify that similarity in advance. So before I even build a model, I think about the phenomena that I'm interested in the wet lab system and the phenomena that I need to generate from the analog. And I even say, what level of similarity will I be happy with? If in the wet lab, you can only get within a factor of 2 by replicating the experiment, then that says something about the level of similarity that you'll be happy with during the simulation. So I would like to come up with a measure mapping model to tie those two things together and establish a degree of similarity. Once I've established a degree of similarity, I've achieved a degree of validation, and I can say that there may be counterparts in the biology to the analog mechanisms, and the reason is I'm matching up some phenomena, and I'd like to iteratively improve that process. If I can do it both at a high level and at a lower level simultaneously with the same analog, I've improved my confidence in the entire analog. And what's more important, I'll get to that in a minute. What's more important is that if I do it at a high level and at a low level, I can look at the analogs mechanisms in between, and there may be counterparts in the biology, but by looking at the analog components in between, it might help me identify something that I could actually test in a wet lab experiment. And that very focused test, no matter what the outcome, falsification or not, would give me new useful information. With the kinds of analogs that I've been talking about, a very nice property is that these components can operate at different speeds, and that increases the flexibility of mapping between phenomena in the simulation and phenomena in the wet lab. I'm told by my wet lab colleagues that they see evidence in the wet lab of cell types in the same system that seem to be running at different speeds. So we would like to have the ability to have components within analogs run at different speeds. So how would you actually parameterize an analog like the one that I just described? Where do you get the parameter values from? And how do you get the parameter values in the face of pervasive uncertainty and persistent and intractable experimental variability? It's easier than you think. I'll give you a very simple example. Human alveolar type 2 epithelial cells are frequently isolated from lungs that don't get transplanted at UCSF. And those are used for studying a variety of things, asthma and so on and so forth. And we have in vitro cultures that they're used to create organoids, and those organoids are then used for experimentation. If you take these type 2 epithelial cells and you put them in 3D culture at different densities of cells, they'll all form structures, but the pace at which they form structures is different, and the kinds of structures they form are a little different. The cells move around, even though they're primary type 2 cells, the cells move, but they don't always move the same way. And also the structures move. And it gets kind of complicated. And there's a lot of theory out there about chemotaxis and this and that and so on and so forth. And all we wanted to do is to come up with a simple parsimonious analog that could quantitatively describe the phenomena that were being observed in the wet lab. So we have 3 spaces: cells, luminal contents, and media. That's all we need in terms of spaces. And cells move, and the cysts move. So it's not a very complicated system. If we discretize it at the level such that a cell occupies a single spot on a hexagonal lattice, I can account for the 3 kinds of spaces and also cell movement. And when I do this diagram on the top, the dark spaces are lumen, the white spaces are media, and the gold and blue things are cells. And in the top panel, it shows you a situation where the center cell is surrounded only by one other kind of object. So on the left, it's surrounded by other cells. On the right, it's surrounded only by lumen. And in those situations, it's likely that the cells will do different things. And these other patterns show other kinds of arrangement. So there's a finite space of arrangements that cells can find themselves in. And the next thing to do is to come up with some likely behaviors of those cells. And that space is finite because of the way we've discretized it. And these are some of the behaviors that they can come up with, but I'm not going to take time to go into that right now. But we can come up with a finite set of behaviors. And what we can do is we can randomly assign any one behavior to any cell in any particular situation for any given cell cycle. So we don't make any decisions. We just say they're going to behave in one of these ways. And when we do that, we get a bunch of crazy phenomena, all kinds of weird behaviors. So the weirdness tells us that we can shrink the parameterization space by eliminating abiotic behavior. We can reduce the number of options that we give the cells in any one cycle until we begin to eliminate any behavior that's not seen within the cell culture. And because we have a lot of phenomena to look at, size of cysts, speed of cells, speed of cysts, how quickly they form a lumen, how quickly they clear out a lumen, because we have a lot of phenomena to look at, we can parsimoniously and incrementally put more and more phenomena pressure on the space of mechanisms and reduce the parameterization down. And we did that, and we were able to match quantitatively the data across a large number of attributes. And in doing that, we found that the behavior options that we gave the cells, for any one condition, the cells chose only one behavior 95% of the time. So the cells had a very good idea of what they wanted to do in any particular situation. And we didn't have to make a parameterization decision in order to get to that point. So that's the strategy that one can use to parameterize a model like this. In essence, we start with a huge mechanism space, and any mechanism in that space has a large parameter space. We can shrink both spaces by eliminating abiotic behavior and abiotic parameterizations. And then we begin the process of iteratively trying to shrink both. We take a mechanism, select a parameterization, ask it if it has achieved our similarity measures. If yes, we're happy. We go back and repeat the process. And if no, we shrink the parameter space a little bit more and continue that process until we toss out that mechanism as being infeasible under these conditions or as being possible. We always follow very strict parsimony guidelines. We try very, very hard not to make the models any more complicated than they need to be in order to explain the phenomena. And whenever we increase the granularity of a model for whatever reason, that expands the mechanism space, and we have to be careful that we don't overdo it. When we revalidate, we always ask, can we simplify the now complicated model and make it a little bit less complicated? So we adhere tightly to this parsimony guideline. And I think it's obvious that in order to make progress in doing this sort of thing, we need automated methods. We did that by hand, what I just described. Oh, okay. But everything that we did could have been automated. So if someone were working in the software environment and understood how to do that, they could have looked at the records that we kept in going through that iterative procedure and come up with an agent that could have done that entire parameterization process automatically. And that's an important direction that I see the whole field going. And once we've successfully achieved a bunch of validation targets, then we pick another attribute, add it to the list. Instead of 5 attributes, we now add 6. Usually, adding a new attribute falsifies that mechanism, and we have to do something, increase the granularity of the mechanism in some way, and then repeat the process so that we still achieve the 5 validation targets that we had before, plus the 6th. And again, you can see that it would benefit from having automated methods that would allow that process to move forward. I envision by the time the students' careers are half over, that process will be automated, and that automation process will discover mechanisms that don't make sense, but withstand an awful lot of falsification effort. It doesn't follow that every mechanism that biology has is necessarily logical to us. So the automated methods might uncover some very interesting biology. Following a rigorous protocol facilitates generating multiple mechanistic hypotheses and then eliminating those that are least plausible through falsification using not wet lab, but in silico experiments. So in that example that I gave, all of our falsification efforts were done through in silico experimentation. An iterative model refinement protocol is I believe, at the heart of this mechanism falsification, this discovery process. Now I'll take a couple of minutes and just give you 3 examples of the more complicated kinds of models that we've been working on using these same methods. I talked about the type 2 alveolar primary cell forming cysts. Some of you are probably familiar with MDCK cells. A couple of my colleagues use in vitro epithelial cells to explore the properties of epithelial cells and have some very nice in vitro methods for growing organoids. They use MDCK cells, primary breast stem cells, so on and so forth. In the case of MDCK cells, this is a picture of what happens over the course of 10 days in a very biomimetic 3D cell culture. The MDCK cells move around. Sometimes they form clusters. At some point between day 1 and day 2, they begin to polarize. As soon as they polarize, they start to form a lumen. They continue to divide. They re-engineer the lumen space and expand. And eventually, they form a single layer hollow cyst. And the size of that cyst kind of doesn't quite level off, but it starts growing very slowly. Because once it gets to a certain size, the cells know they've formed a proper size lumen, and so they're more interested in re-engineering the lumen contents than they are in forming a bigger lumen. In the process of doing that, they make mistakes. So that little arrow indicates that sometimes they try to make an extra lumen where they shouldn't. Or instead of forming a nice single layered cyst with single cells, sometimes the cells bunch up on one side. So they misbehave on occasion. And we have quantitative data for this entire process. It's been repeated separate experiments, time-lapse images, lots of data. So what operating principles are each of these cells using at each stage in this process? We should be able to get a handle on that. Once we have a set of operating principles, once we know what the cells are doing at any one time, then we have a plausible hanger for looking at more details. But we need to know what the cell is trying to do in order to have a context for fitting in subcellular molecular level mechanisms. So that was the first task. So how and why do these cells do what they do? What are the cell's operating principles? What happens when an operating principle is disrupted by a molecular intervention? Those are the questions that we're interested in. So we completed the process that we described before, except now at a higher level of resolution. And we were able, again, to achieve quantitative validation targets across a large number of attributes. And remember that a regular hexagon and hexagonal space, each of these little dots is a hexagon. So this is a very fine-grained hexagonal space. Each of those dots is a regular hexagon. So a regular hexagon in hexagonal space maps to a circle and continuous space. So those hexagonal cysts are actually quantitatively matched to spheres. Well, we've put a lot of energy into a multi-scale model of the liver. I don't have time to go into it, but again, it's components with inside components with inside components, nested from the organ level all the way down to the components with inside hepatocytes. And we're interested in adding compounds, natural or xenobiotic, and looking at the consequences. An obvious use would be to look at hepatotoxicity of compounds. Another use would be disease progression, repair, tissue regeneration, etc. We would like to use the same model for all of those in silico type experiments. The advantage of the approach that I've just described in this context means that I can take one normal in silico liver model, revalidate it for 2 different disease conditions, and I know what in silico what was required to change the model from normal to disease 1 and normal to disease 2. So I have a hypothesis about what might have actually transpired. In the wet lab, it's almost impossible to do experiments on normal systems that are in the process of transitioning to model disease systems. So having that in silico capability is useful. And we can hypothesize that there might be multiple levels of similarities, both in transition and in terms of mapping from in silico to wet lab. So this picture illustrates where we would like to go. One of the more challenging sets of modeling experiments had to do with leukocytes. You take leukocytes from mice and you put them in an in vitro system, flow system under a microscope. They always behave very individualistic, but their behaviors are confined to a finite set. You can't predict what any one leukocyte is going to do, even halfway through the observation. So can we come up with mechanisms of behavior for the leukocyte that validate against the behavior of individual leukocytes, and then at the population level? And then further, can we take data from genetically modified leukocytes that have differences in terms of their cell surface properties, ligands and receptors, use that same leukocyte, revalidate it against other data, and build a model that allows us to link population level behaviors all the way down to what's happening at the cell surface level when ligands are diffusing around and forming clusters or not. So we were able to do that with a set of observations. And in this case, the cell is an object divided into a large number of quasi-autonomous units. And each of those units is represented by a quasi-autonomous piece of cell. And inside those are other units. And then finally inside those are other units. And in the far right-hand side, within each small time step, those objects actually map to individual molecules, and they're diffusing around and not forming clusters or forming clusters, and that's having an impact on whether the cell adheres or not adheres or whatever. So sorry, no equation. But the phenomena match very well. What this does is it says that we can hypothesize that a lot of the behavior of a leukocyte in terms of interacting with the surface is a result of quasi-autonomous units within the cell doing their own thing, independent of what the other units around it are actually doing. I don't know if that's right or not, but we were able to use that hypothesis and come up with some and validate across a large set of data. Going back to the take-home messages, discovering and developing new therapeutics requires acquiring and exploiting deep insight into the complex mechanistic networks that are responsible for the phenomena of interest. And doing so in the face of incomplete data, multiple uncertainties, requires building and challenging better mechanistic models. The methods that I've outlined can facilitate accrual of validated mechanisms, help form concrete hypotheses for translation, and bring into focus concrete details like I just showed. Is it necessary for those molecules to form clusters in order to get the right kind of adhesion? We can test that type of hypothesis. In silico, you can't test it in vivo. Can bring into focus concrete details for particular innovations and increase the ease with which accumulated mechanistic knowledge is measured, visualized, and leveraged. End.
Does anyone has any questions?
So I hope I've aggravated somebody at least well enough so that they'll ask a hard question.
Right. Yeah. I just wanted you to continue on that last slide. I mean, where you said you wanted to come to, so to speak, the next step. come to some validation and to a new level of understanding, whether you could go back to the leukocyte example and say, quite how far you got, whether you carried all steps out of the 6-step process you were describing.
We did. We did. And in the process, we learned a lot about the methods that we were using. And one of the lessons that we learned is that we have to adhere very tightly to good laboratory practices doing the simulation work. And the reason is that if we take this validated leukocyte and we now increase its granularity in order to validate against this data, and now we take it over here and we try to validate against this data, usually we'll find out that we need to back up a little bit, go back over to this original one, and then redo it with the notion of coming back over here. If we don't keep really good records of what we did, it slows us down. So we learned a lot of things in the process of doing this, some of which I think will help in doing automation. It taught us some good lessons about expanding mechanism space as we increase granularity, etc. But this data comes from strictly in vitro, where the cells are isolated and flow through a chamber, in situ, where fluorescently tagged cells from one mice are added to another mouse, and a vein is isolated, and you observe the fluorescent cells moving through and actually moving through a vein. And then taking those same cells and putting them in another mouse where the endothelial cells have different adhesion molecules than the first set. So it went across multiple experimental conditions and taught us a lot about the need for relational grounding in order to move the same analog around those various experimental conditions. I saw you had your hand up there earlier. No?
Mechanisms involve complex sets of interactions. Might it be possible to actually measure the interactions over time before doing any modeling?
Well, that goes back to my one-to-one mapping, quantitative mapping. If we always insist on having more information before we create the model, then the models will always lag behind the wet lab. And what I think what we need is we need models that are sufficiently good and sufficiently adaptable so that they can run simultaneous with the wet lab. So my answer to your question is no. Don't wait till you get more data in order to try to do a model. Try to figure out a model method that'll work for the data that you have. Yeah, please. I'm trying to be provocative to generate some more good questions here.
The thought behind my question is that those measurements might be measuring an emergent system property, which itself then could then become an object of scientific study.
You're talking about the wet lab measure or the in silico measure?
For example, if you have two proteins, two hormones that might interact over time, for example, or brain regions that interact over time, or any component, such components that can interact over time, I think it is possible to actually measure those interactions over time and have a measure of an emergent system property that then becomes the object of scientific study before any modeling, but then you can model those interactions over time to have a more complete systems perspective.
I appreciate that. I realize that that's a perspective that's held by a number of people. And I encourage that approach going forward. However, in addition to that approach, I think it's also useful to have modeling methods that don't wait on additional data in order to explore those same phenomena. For example, if you take multiple sclerosis, the mechanisms behind multiple sclerosis are really poorly understood. Does that mean that we can't begin to model MS now? I would say there's plenty of data out there about MS in patients that could be the object of modeling and simulation experiments now, but it might be pretty crude, but they could be used now. So I think there's room for multiple approaches, but I would not encourage anybody to wait to get more data, especially with something along the lines of an emergent phenomena, because emergent phenomena depends on the aspect of the system we're looking at and the perspective taken by that measurement. And that's just my opinion. If you want to describe the pattern in the data, that's one thing. I'm not talking about describing patterns in data. I'm talking about trying to better understand the actual mechanisms as opposed to describing the pattern of the phenomena. I want to get at the mechanism. And the only way I can shrink the space of plausible models is to have a method to falsify some of the models that populate that space. I have to be able to falsify them. Otherwise, that space remains large. And it can't be sampled using wet lab experiments. It's too big. There has to be some way to shrink it. And I think that's part of the reason that there's been such a problem with the pharmaceutical industry. When I've consulted with them, it's very, very clear that they're not doing a good job of shrinking plausible mechanism space. They're actually adding to it. Well, we have to have methods to shrink that space, otherwise I don't see how we can make the kind of intervention progress that we want to make.
Actually, so this is a little bit of a refinement on the other things. What about, so especially if you say, you know, confronted with initial condition or data variability or, you know, inaccuracy. So can you apply in your scheme of things here some, you know, physical or engineering principle like stability? And, you know, do you recognize a mechanism for being stable or not stable beforehand? And is this,
Okay.
you know, this might be sort of an a priori criterion. You know, if it works biologically, then obviously it somehow is immune to certain kinds of perturbations of conditions.
That's a good question, and it comes up when you have experiments that are done in vitro over a relatively long period of time, because you wonder if the mechanism is stable or is it changing over time. So that taking off my biology hat and putting on my engineering hat, the first thing that I would say is, can we come up with a plausible explanation that does not require change? And then can that withstand a couple of rounds of falsification effort? If it does, then we don't need to go down the mechanism change route until we're confronted with some observation that gives us no alternative. But the stable mechanism is a simpler explanation. So I'll stick with the simplest explanation for as long as it will survive. That's how I would answer that. One here. Hopefully a skeptic.
No, not skeptical. I don't have enough knowledge to be skeptical. Actually, two questions. You have a liver model up there, and it's been a while since I've looked at it, but Imran Shah, I believe, and John
at the
Weibaum,
EPA.
at the
And
EPA.
also a group in Germany have done other models.
Yes.
There are a lot of liver models out there.
And do they subscribe to your thoughts and methods?
I interact with Imran kind of on a monthly basis.
Mm-hmm.
And I've noted incremental stepping in my direction. But in terms of the German group, their focus is somewhat different. They're very focused on tissue regeneration. And the thought leaders in that group all come from the physics domain. And so there's a bit of a hostility about the notion of relational grounding. So that's fine.
Okay.
I think it's good to have an interface there to discuss. And as I tried to point out on those scales, I think there has to be a wide variety of different modeling methods available in order to cover those scales. And right now we only have a few, and we need more modeling methods. We need graduate students to think of entirely new ways of going about modeling some of these systems. And that'll contribute then in the future and switch from modeling method, depending on what the use case is. If the use case is very focused on something that might be mechanical force driven, then you might need a much tighter linkage to absolute grounding.
And just a follow-up question, you had a slide which had various time scales for
Yes.
cell and tissue, etc. Have you run into any models which you've created which really have such a fine mechanistic action that you can't compute it in?
Yes, the leukocytes.
Yes.
In those models, when our simulations move forward in simulation cycles, and the logical thing to do is to make a simulation cycle map to a certain number of seconds or minutes. And it turned out that for the leukocyte model, we ran up against a brick wall until we made that mapping flexible, so that early in the operation of the mechanism, the mapping from simulation cycle to the time scale was, let's say, 1:1, and then at the end it was 1:2. So by having that flexibility, it allowed us to move further, faster in that model. And that was at the whole cell level. In the ratio of time steps at the cell level to small steps at the lower scale is 1:10. But in wet lab time, it's 1:1000. So the coarse grain mechanisms, their mapping to time is one thing, and it's different at the coarser grain. If I insisted that it be the same, it might force us to make an entirely more complicated system. So allowing for different parts to operate at different time scales gives us flexibility to use a more parsimonious mechanism to explain a larger set of observations.
Got it. Thank you.
See on those two time scales in vivo.
Oh, you mean with in vitro whole animal data?
Yeah.
We're working on that right now. In the case of compounds like acetaminophen that cause hepatotoxicity, the pharmacological, the toxic phenomena tend to emerge over different time spans, and so we're looking into ways to change the mapping between cell simulation cycles and wet lab time in order to deal better with these different emergent properties that come out at different times. And we're doing that right now. So I think it'll be successful, but I can't say that it will be successful. But that's our vision of how to deal with mechanisms that are taking place where every second counts. And then we're actually interested in, well, what happens at 2 weeks? And we would like to be able to run those simulations, and we can do it if the mapping doesn't have to be constant.